Hello, my name is Rhea Kanwar, and I'm going to be doing a presentation on a very interesting cutting-edge research study. The title of the research article is Double Cortin Positive Cells in the Adult Primate Cerebral Cortex and Possible Role in Brain Plasticity and Development. The picture on the slide is of double cortin positive cells. I will be discussing these cells later on in the presentation and describing their location as well as their function. To begin, the general purpose of the study was to determine the expression of DCX in the cerebral cortex of the adult human in two species of monkeys. To give you a brief overview, the lead researcher of the study, Joycelyn Blouch, was a functional neurosurgeon who began studying this 15 years ago as a chief resident. She was working days and nights in the emergency room and often had to take care of patients with head trauma. When a patient comes in with severe head trauma, their head is swelling, and to decrease the intracranial pressure, you have to sometimes remove a piece of swollen brain. So instead of throwing it away, she decided to study them in hopes of finding a way to repair the brain. The research study is important because it gives insight into double cortin positive cells, which have not been studied till recently. It is also important because it could be the answer to repairing the brain. Currently, one of the famous surgical strategies for neurosurgeons is called deep brain stimulation, where you plant an electrode in the depths of the brain in order to modulate a circuit of neurons to improve a neurological function. It is a really amazing technology in that it has improved the destiny of patients with Parkinson's disease, with severe tremors, and with severe pain. However, neuromodulation does not mean neurorepair. The three main research questions for the study were: are cortical autografts a useful therapy for monkey models of neurological disease? What will happen if double cortin positive cells are reimplanted into a normal brain? And what will happen if double cortin positive cells are reimplanted into a lesion brain? Below are two pictures, one of a normal brain and one of a lesion brain. This study used qualitative data. This is because they used observation to observe the monkey's performance in the food pellets. They used observation to assess speed and accuracy differences. The color, shape, and function of the double cortin positive cells were described. This study was based on tissue from human and non-human primitive brains. Pieces of the cerebral cortex and brain was taken from 3 adult cynomolgus monkeys. Biopsies and brains were taken from 2 adult African green monkeys. Pieces of 6 brain resections from human neurosurgery was taken from trauma and epilepsy. Next, I'm going to give an overview of the procedures done for the study. The idea was to biopsy a piece of the brain in a non-eloquent area of the brain, and then to culture the cells. They were then labeled by putting color in them in order to be able to track them in the brain. The last step was to re-implant them in the same individual. This is called autologous grafts or autografts. This means that they wanted to grow cells from these pieces of tissues. It was not an easy task. They needed the right nutrients, the warmth, and the humidity, and all the nice environments to make them thrive. Some of the main findings in the study was that it was determined the expression of DCX in the cerebral cortex of adult humans and 2 species of monkeys. 4 different populations of DCX positive cells were discovered. It was determined using mRNA and protein quantification that DCX was detectable in all of the human neurocortical tissues. It was determined that the 4 DCX positive cell populations were co-localized with different neuron and glial markers depending on their location in the cortex. Lastly, to conclude, this is my citation for the study I presented on. I found the study very interesting because I think it could be the answer to brain repair one day. I think that they were very creative with their empirical paradigm with using monkeys and using their knowledge as chief residents to continue the study. Although it was a very difficult article for me to understand because I'm not very familiar with many of the procedures, terms used, and testing, I'm glad I shared this article because I learned a lot from it. I want to pursue neuroscience research in the future and hopefully do something with stem cells.